All right, so we're doing active transport. Specifically, we're going to talk about endocytosis, exocytosis. It's really not that hard. We just need to kind of get a couple pictures and fill in some blanks in your notes. Otherwise, um, there's only a few things to deal with anyway. So we can get started right away here. Your objectives are to describe active transport. Last section we did passive transport that required no energy. Active transport just means it uses energy. It actually costs your cells to move things across the membrane using active transport. Then we're going to talk about endocytosis, phagocytosis, exocytosis. Other than being hard to say, they're not that complicated. We're going to try and draw a picture to represent those so you can see how they work. But otherwise, I just ask questions if you're not quite understanding it, because it is kind of hard to draw the pictures and maybe you don't understand it right away. But it's not too bad. Okay, first off, active transport. It's bigger. Moving molecules against their concentration gradient is going to require energy. Remember before we talked about letting things go where they want to go? They always want to go from high concentration to low concentration. This is when you want to force things to go against their concentration gradient. So you're moving them from low concentration to high concentration because you need to do that in a cell. You need to be able to get lots of the things you need into a cell. You need to be able to move things from one side of the membrane to the other, right, for a lot of different purposes. And to do this active transport, we're going to use protein pumps. So you're going to draw a picture in your notes to represent this. You just need a generic membrane. Remember, you can draw two tails for every phospholipid, but I don't really need to do that. And you're going to have a protein in there that started in this conformation. So you can see it's closed at the bottom and it's open upward. Right? And what's going to happen here is I'm going to have some kind of ion, maybe a sodium ion, on this side of the cell membrane that I'm trying to push to the other side. So this protein is open right now, and this sodium is going to make its way into there randomly. And when I get sodium into that protein, that's when I'm going to do the work. That's when I'm going to change things. So again, it's kind of hard to animate in notes. But essentially, I'm going to take this protein that's open upwards, and I'm going to use energy to cause it to flip so that it's open at the bottom instead. So I'm going to change the shape of this protein so this new picture is the same protein, just flipped, so that it's open down instead of up. And what that does is it forces that sodium to exit on that bottom side or the other side of the membrane there. I can finish my membrane here. But you have that change in conformation, this shape open upward to open downward. That costs the cell energy. It takes energy to make that protein change. And the whole reason for this, remember, if that sodium wanted to go across the membrane in that direction, you just give it an opening and let it go across. We're forcing it to go against its concentration gradient. So we're building a concentration down here. This concentration is getting bigger and bigger and bigger. And this concentration out here is getting smaller and smaller and smaller. If I was just to open a protein channel, this would actually want to travel this way across the membrane, but I'm forcing it to go down instead of up. Everybody kind of see what I'm talking about there and how that's working? You got a protein that's open like this. It catches stuff from the top side of the membrane, and then it just changes shape and pushes those things out the bottom. Best way to model that, at least. And I'm going from low concentration to high concentration. I'm trying to build a really high concentration of sodium on one side of the membrane. And because your cell actually likes to not waste energy, because it costs you energy to change the shape here, we're probably going to have a dual function for any given protein like this. I might also have a different ion that I'm trying to move. Maybe it's potassium. But I don't want potassium on this side of the membrane, I want it on this side of the membrane. So when this protein is open downward in this conformation, it's going to collect potassium ions, and then you're going to flip this back to this conformation because it's got to be open there again anyway to collect more sodium. But when it flips back, it pushes the potassium out this side. Right, so not only would you be moving one thing against its concentration gradient, but you're probably going to move something else with the same protein because it just makes sense to save some energy in that. Right, if I got to flip this back, but there's no reason to flip it back other than to collect more sodium, it would be a waste of energy to have a whole nother protein pump that's flipping back and forth to push potassium across the membrane. But again, I'm pushing these things against their concentration gradient, so obviously I'm trying to build a high concentration of potassium on that side of the cell membrane, and a low concentration on the other side. And your cells are doing things like this all the time. In your cells, you have potassium, sodium pumps, and they don't actually send, 1 for 1. It's actually like 3 to 1. And what it does in the long term is it builds an electric potential across your cell membrane. So that at any given moment, I could take another protein channel over here, open it up, open up the floodgates, and sodium starts rushing through one way and potassium starts rushing through the other way. And when you do that, it can actually send a signal from your brain to your toes to actually make your toes wiggle, right? You have to be able to communicate with your cells, and you do that through electrical potentials. And you can only build those by forcing ions to go across the membrane and building up concentrations on each side. And once you open those floodgates, like I said, then they just start passing through one way or the other, and that's when you send that signal from your brain to your muscles to make them move. Right? You guys have probably heard of those ions in your system inside your body being called electrolytes, right? And what are you supposed to drink to replace those electrolytes in your sports when you're playing? Gatorade, right? That's what it's trying to sell you, is electrolytes. Ions that need to be in your system. Why do you need those ions? What's one thing that can happen when you're playing football or basketball or volleyball or whatever?
Cramp.
You can cramp. And a cramp is essentially when your brain loses that ability to tell that muscle cell to stop tensing up. And that might be because you have a lack of some ion or another, so you can't make those electrical potentials, so you can't actually signal and tell it to stop doing that. So it gets stuck in one position or another. So there's a reason why you need to replace those things. Now that being said, if I talked about it in a biochem class, I'd probably tell you that most of us, working at a high school level in any sport, you're probably not losing a significant amount of electrolytes that you need to replace them with Gatorade. Now, Gatorade still tastes good, and it's got the sugar in there to give you a little boost of energy too. But that being said, you could probably make a glass of Kool-Aid and then put a little salt in it and drink it. It would be about the same thing. That's basically what Gatorade is. But professional athletes do need that because they actually literally work out for a whole day, like 8 hours at a time. And, they're pushing their bodies to extreme limits. They need to replace that stuff. You replace it every day with your diet as long as you're not just drinking Mountain Dew and eating Doritos. Right? If you have a normal, balanced diet, you have plenty of electrolytes. You probably don't need Gatorade, but you need those things. Right? So again, this is supposed to be a pump. I'm forcing the sodium against this concentration gradient. I'm forcing the potassium against this concentration gradient. You build up those electrical potentials. You can use that to signal from your brain to your toes and tell them to move. Okay, so this question down here. I think this is the only real whiteboard question we got today. Why is energy required in a protein pump? What do I use that energy for? There are two things I want you to mention in this answer specifically, so try and get something written down. I'll check on it and see if it's okay. What is that energy used for? Okay, so part of your answer has to do with the protein itself. Part of the answer is from what we were talking about before and how we didn't have to make those things move yesterday for a reason. So I actually want a more specific answer. What is the energy used for? Because you're saying the right things to a certain extent, but why would I have to force that sodium to go to one side and the potassium to the other? So why wouldn't they just want to go that way? So I want that part for sure. And then another thing, what did I use the energy for? Just physically speaking up there. What did I have to do? So this is one of those questions kind of hard to answer, but if everybody participates and you write stuff down, I can probably pick from this board and that board and this board and get a good answer overall. I kind of like what Dylan has and Aaron had, right? Aaron, what did yours say?
Moving molecules against their gradient.
So you're going to move them against their gradient, and Dylan used a specific word that I kind of like saying in that. You're going to say what?
Push.
Push, right? You're forcing them to go where they don't want to go. Yesterday, we just let them go through, right? We just opened up a channel, goes through. Today, we have to force them to go through. That's the difference between that facilitated diffusion and this protein pump. I'm forcing those things to go where they don't want to go. I'm pushing them against their concentration gradient. So that's one thing I want you to talk about there. Using a word like that is actually pretty good, a good idea. Say push or force them across against their concentration gradient. But how am I going to do that? That's the other part of what I use the energy for. And I think they kind of had a pretty good answer for that. What was the other thing aside from the concentration gradient that you said?
To switch which way the protein is open.
You got to change the shape of the protein, right? And it's going to cost you energy to do that. So flip-flopping it back and forth, that's what the energy literally goes into, is flipping this back and forth from open one way to the other way. In the process, you're going to push those things against their concentration gradient. But, so talk about how you change the shape of the protein. Right, you'd need a complete sentence. Talk about how you're pushing those ions against their concentration gradient. It wouldn't cost me any energy at all to just open it up and let them go where they want to go. But because I am not trying to make them go the way they want to go, that's why it costs me energy to force them to go that way. So those two different parts. As long as you answer both parts, you would have the complete answer and get full points on your quiz, on your test. And yes, this question will probably come up on your quiz that you got tomorrow. So make sure you have a good answer there. Okay, otherwise we have endocytosis, phagocytosis, and exocytosis. Again, other than being hard to say, they're not really that complicated. Especially if you remember from before. What word did I want you guys to think of when you saw endothermic? When it was endothermic, did the energy enter the system or exit? So if it's endocytosis, are those things going to come into the cell or exit the cell? And if you think of this word when you see endo, you'll remember which one's which. Large materials are going to be enclosed by the membrane, transported by vesicles. Right, I'm going to try and draw this to give you an idea of what it's supposed to look like. It's not the easiest thing in the world to draw. It's actually fairly easy to understand just from a common example, real world example. But something is going to enter the cell. So I have a cell here with a membrane. And I have this thing, green, that I want to enter the cell. So first, obviously, that's going to need to come near the surface here of the cell. But then the next picture, what's going to start happening, so I'm going to start pushing in on that membrane to start to form a little vesicle. Right? And these two parts here of the membrane need to come together to fuse, to join, to make that little bubble within the bigger bubble. And then your last picture, you have that little vesicle inside the bigger cell that's got the stuff that you need in it. So you just create that little vesicle, you create a bubble within a bubble. I wonder if I still have that over here. You guys have probably seen this kind of phenomena before. Like bubbles, right? There's a membrane there. And actually soap is a fairly good representative of a membrane. Right? Each one of those little bubbles was like a little cell. And that soap was actually a lot like a phospholipid. There's polar, nonpolar parts. That's why that bubble forms to begin with. But as I'm forcing air through this membrane, the membrane is going to enclose on itself and form little bubbles, right? So the air entered this side, but it went through the membrane and it made little bubbles, little vesicles on the way through. Does that make sense? So it's hard to animate and it's hard to draw, but it's an idea that you've seen before. You know what that kind of looks like, right? That membrane closes in on itself and you make those little bubbles inside the bigger bubble at the end of the day. So that thing that you needed to be inside the cell is now inside the cell. It's just inside a vesicle inside the cell. So that is endocytosis. If it's phagocytosis, that is just a specific kind of endocytosis that's used by white blood cells in your system. So your white blood cell is going to engulf some foreign body, whether it's a bacteria cell or whatever. And it's going to digest, destroy that thing so that you don't have to worry about it anymore. But it's going to have to swallow that thing first. It's going to have to take it in first. And it does that through endocytosis. So endocytosis, phagocytosis, really the same thing, more or less. It's just phagocytosis, you're specifically talking about engulfing foreign bodies and digesting them in your white blood cells. And so again, hardest thing about these is actually trying to say them. Can you say that word, Enrique? Endocytosis, right? It's not that hard to say, but it's somewhat difficult. Nate, can you say that? This word right here. Say it a little louder. I didn't hear you. No, the whole thing. Endocytosis, right? It's not that bad. This one's a little harder. Can you say that one, Francie?
Phagocytosis.
This is just an F, essentially. Right? Phagocytosis. Dylan.
Phagocytosis.
Right? Not that hard to say. So you could probably pull it off at some point or another. Right? If you have to talk to each other and tell each other answers during a quiz, you got to use that word. Actually say it. But again, you're going to engulf that foreign material and try and digest, destroy it. Exocytosis. Remember when you see exo, I want you to think exit. So you're trying to get stuff out of the cell that shouldn't be in there anymore. So it's a waste product, probably. Sometimes it's not a waste product. Sometimes it's something you need to secrete, like a hormone or a mucus, right? The cells inside your nostrils produce mucus that they excrete, and that's the snot that you have to blow out every once in a while, right? And that's necessary. It's used for a purpose. In that case, it was to try and catch things that you're breathing in and keep the bacteria and stuff out of your lungs. But hormones that are secreted in your body, right? Sometimes you need insulin in your blood, so that's going to come from your pancreas, get secreted directly in your blood, and then flow around and help bring sugar into your cells. So exocytosis is not just to get rid of waste. There's also other purposes, but this is how you get stuff that's produced inside the cell out of the cell. And the picture that you would draw for this is basically just the opposite of what we had before. Now I have some waste material that I need to get out of the cell, or maybe it's a hormone that I'm trying to get out of the cell and send somewhere else in the body. So I need this vesicle that it's already contained in to get close to the surface. When it does get close to that surface, it can fuse with the membrane and start to open up. So that ultimately in that third picture, I can expel that thing, whatever it is. That bubble inside the bigger bubble becomes part of that bigger bubble, and you just kick that thing out, whatever it is. You've also seen that kind of action happening before, one bubble fusing with another one, right? You've seen that all the time with bubbles in soap when you're washing something. The smaller bubbles start to become bigger and bigger and bigger, right, as they fuse together. Only this time, you got the bubble inside the other bubble, and it fuses with the other one, and it spits its contents outside. Right, so you need to know the difference between endocytosis and exocytosis. Both of these processes are also active. It takes energy to do this, because the cell, that cytoskeleton that we talked about before, right? It's going to have to start to move to help pinch off that membrane to form this new vesicle. And it's going to cost you energy to move to do that. So, and this as well, fusing this to here, getting this from here to the surface is going to cost some energy. It's going to move along that cytoskeleton. And then it's going to get ejected by fusing with that membrane on the outside. So these would both cost you energy, but you need to do that for different purposes. All right. Everybody good with all their notes? Got everything in there? Any questions?